medical physics and biomedical engineering here. And I'm really pleased to invite you all to Madison. Hopefully we'll have really great weather for the next 5 days. I'm also co-chair of Biomedical North America, which is why we're all here. And hopefully you've seen our very talented illustrator that used the badger. In case you're curious why it's not the Wisconsin badger, we're not rich enough to pay the fees for that. So this is the non-University of Wisconsin badger, but still a badger. And the reason we're the badger state is because we used to be a big miner state, and they used to call the miners burrowing into the ground. In case you're curious, why so many badgers? So we are the badger state. So I'd like to welcome all of you, both in person and remote. If you have any questions, you see a red lanyard, we're here to help. There's a lot of us. So you'll see at least, 10 or 12 or more people in the audience with that. Ask any of us for anything, where the bathrooms are, where the coffee is, where restaurants are, anything you like, just ask us. We'd love to help you. And we'll be there all week. Special thanks to Nikki and Vanessa that did all the work. I'm just here to figure out, so thanks to everyone. And then to our many volunteers that you'll meet this week, involvement. And then thank you to all at BINA, including my co-chair Claire Brown, who's in the back there, who will be, the next one will be at her place. I won't steal her thunder because we'll do it at the end of the meeting, but we are going to have another meeting, and that'll be in Montreal in a year. And thanks to all the BINA Executive Board that I think is here in entire force, which is awesome. I don't think we've ever met all of us together, so it'll be great in person. And all the many working groups. And please talk to anybody in BINA if you have any ideas or questions. Hopefully not complaints, but suggestions. But if you have complaints too, please come see us. And then the many folks locally that funded us, we're very lucky to have academic partners. Center for Quantitative Cell Imaging, the College of Engineering, Office of Corporate Relations, McPherson Irish Institute, the Morgan Institute for Research, which is the home to BINA, and the SP Optica Student Chapter all have contributed to making this meeting happen. And then, of course, Chad Zuckerberg. We're very, very grateful. Chad Zuckerberg has made BINA happen. More than just a funding partner, they've been intellectually involved with regular monthly meetings, giving us great suggestions. So we're very grateful to really see that being the inaugural partner of BINA. And then the many, many sponsors, most of them here in person. I won't have time to go through all of them, but just really grateful for all these great companies that not only are sponsors of the meeting, but are involved year-round in special events, including career and commercial events. So very grateful for all these companies, and please come thank them if you see them, and go to their tables. Many of them are here in person. And now, Vanessa's going to say a few things.
All right. Hi, good morning, everyone. A little slight change of plans that I'm up here right now, but figured to go through some logistics really quickly. So as Kevin said, I'm Vanessa. I'm based here in Madison, Wisconsin. Welcome to the Badger State. We're very glad to have you. Reminder about our code of conduct for this meeting. So the behaviors listed at the top are things we strongly encourage. Those listed at the bottom will not be tolerated, and if you have any issues, please reach out to us at the contact@bioimagingna.org email. So badges, you should have noticed when you came in, there were some bigger badges and some smaller badges. Unfortunately, we were somewhat size limited at lunch, so the big badge indicates that lunches are included. All of you, when you signed up and registered, were able to select what dinners you were attending. That is listed on the back of your badge, as well as the EOEs that you signed up for on Friday. Yep. And then there should have been 2 drink tickets in every single badge, so please join us for the welcome session and the poster session. And as Kevin said, red lanyards. If you find anyone with a red lanyard, please ask them for help. The little BINA logo badges indicate that those are for working group members right now. You guys can be able to collect them a little bit later in the week, but we wanted to make sure you knew who our working group members were and could find them and ask them questions about the exciting work they're doing that you'll hear about throughout the week. If you provided a pronunciation for your name, that is listed towards the bottom of your badge. And different folks have ribbons, volunteers, but if you're a first-time attendee, we should have some badges or ribbons for you on the shared table with materials, so you can collect that from there. And just a couple more logistics. If you're going to be using social media, please tag us and use the hashtag BINA2024CC. Lunches will be 90 minutes, and we will have lunchtime topics that different working groups have suggested to discuss. But also, please feel free to take time for yourself. It's going to be a long and exciting week. We've got lots of really exciting things going on, but we know you all have busy lives back home and you might need to check an email or do different stuff. So please feel free to take a moment for yourself. And also, any evening social activities are entirely optional. So please join us. We encourage you to, but also please don't feel obligated if you have other things you need to attend to. The maps I'm about to show are available on SwapCard and linked from the full program. You should have seen QR codes around the rooms and by registration to access those. So we are here in the marquee. Hopefully you were able to visit Varsity Hall and grab a coffee this morning. The elevator is circled in green and the restrooms are right in between the two of them, as well as a water fountain. And then there's also a water bottle filling station on the first floor if you need that. Please visit our vendor tables. As Kevin said, we're very grateful to have so many supporting us this year. And the topic tables that are listed with the numbers on the lunch tables will be explained later on as we proceed lunch. If you need coffee or souvenirs or different stuff on the first floor, those are available. And on Tuesday and Thursday evening, we will be in the Trip Commons, which is on the second floor of Memorial Union out by the lake. There's a map to show how to get there. Again, linked in all of those documents. And that is my last slide. Nikki will be speaking a little bit later in our next session. So with that, I'll pass it back to Kevin.
And I'm very honored to introduce two great people. One is Mike Davison, who is very good to me, as many in the field. He's just an amazing technologist, and we're honoring him, and we're delighted that Eric Betzig agreed not only to talk about his work, his own work, Eric Betzig's work, but also Mike Davison. So this is the first time we're honoring Michael Davison. We hope to do this in the future. His widow, Pam, is in the audience. We're delighted that she's here. But he was a great person, as Eric will talk about. I can't really do justice to Eric's career. He's had an amazing career. He's currently a professor of physics and molecular cell biology at Berkeley, but very well known for his work at Genelia Farms and also, of course, his Nobel Prize in 2014 for his pioneering work in superres. So we're delighted that Eric can join us, and also delighted that he also wanted to honor Mike Davison. So let's introduce Eric. Thank you.
Cool, thank you.
And let me just make sure that the Zoom is.
Take your time, and you know what? I'm going to talk while she's figuring that out.
I have a lot to say about Mike. So Mike is the only guy I know who has a crazier career path than I do. He got his undergraduate degree after moving around from a couple places at Georgia State. And then he came to Florida State in order to start a PhD in molecular biology. But I think both he and I are fish out of water in normal academia. And so after a few years, he decided to do something and pivot to something completely different. In his case, it was starting a business to plate auto car parts with chrome. And he did that for a number of years. And then in the early 90s, or maybe it was later, or earlier, they had the Florida State won the bid for, stole from MIT, the National High Magnetic Field Lab, which was then brought to Florida State. And Mike basically charged in the door and said that I can help you guys out. And they said, sure, great. And so he started being sort of the staff microscopist to, for example, look at the wires that would go into these magnets, because if they're a very high magnetic field, the force will try to blow them apart. And so you need to make sure that everything is copacetic there. And eventually, you know, just his curiosity with his microscopes got a hold of him, and he started looking at all sorts of things like integrated circuits and so forth, and see little things where the designers would hide like Waldo in terms of being written on the chip and so forth. Had this Silicon Zoo, which gradually grew into his molecular expressions website, which was, I mean, this is like serious cutting edge of web, okay? I mean, he was right at the very, very beginning of this. And grew that, of course, into this enormous useful utility for all of us in this room and anywhere who does microscopy anywhere. Now we're good, okay? So catch up a few slides. Good. All right, thanks. And there's the guy. Man, I remember this exact room when I first visited him going there. So the one thing that he and I had in common, although we didn't know it at the time, was an absolute love of this idea of fluorescent proteins, because I go way back probably into '82, and we didn't have them then, okay? We had dyes, okay? And the dyes often sucked, and they often didn't go where you wanted to go or do what you wanted to do. And so, and furthermore, you know, I was doing near-field microscopy back then, and my dream was to be able to look at living cells with the resolution of an electron microscope. But one of the things were too crappy. There we go. And so when I quit science in '94 and went into the machine tool industry, and when I quit that and wanted to do something different, the first paper I read was this paper from Marty Chalfie, which also came out in '94, which I had just missed. And my jaw was on the ground for a week because of how elegant and beautiful it was to be able to take part of the DNA of a jellyfish, splice it to any protein you want, and get a glowing protein, get the cell to do the work in a live cell and get fluorescence. It was magical. Mike thought it was magical too. And so I wanted to figure out a way to do that. This is before I met Mike. So I started working on, I knew the limitations of confocal microscopy in terms of the out of focus light that has to be stripped away and the bleaching it causes and so forth, and the fact that it's a serial scanning technique. So I started thinking about trying to make a multifocal field, starting from the idea of a standing wave of two coherent waves going together to make this pattern that just goes up, down, and start adding in additional plane waves to create things called optical lattices. And eventually I came up with a theory for three-dimensional optical lattice that would be very dense to try to do massively multi-parallel sort of confocal on steroids to look at a whole cell at once. But I was unemployed. I didn't know what to do. And so I contacted my old friend from Bell, Harold Hess, who I'd been in contact actually for the whole time I was out of science, and he had gone out of science into the disk drive test equipment business. And we both wanted to get back into science. And on an earlier trip, Harold was already very famous for his work with scanning tunneling microscopy in the '90s. And so the head of the National High Magnetic Field Lab was a guy named Greg Bobinger, who was our friend from Bell Labs. And so Greg had been trying to hire Harold for some time, and Harold had made an earlier trip there and met Mike and was impressed by what Mike was doing. And so when Harold knew about my optical lattice idea, he said, I got to take you to meet this guy, Mike Davidson, because I think you guys would get along. And so in April 2005, we went to meet Mike, and I was blown away by everything he was doing. He was just as Harold is a soulmate of mine, the only other guy who was really a soulmate of mine was Mike. We just thought the same all the time. Number one thing of all three of us was hard work. I have never met anybody who worked harder than Mike. And you can see that by a guy who had a whole life and a whole career outside of science and then pivoted on his own terms, not in a university, not writing grants, find a way to do his own science and do it on a world-class level where everybody in this room knows and owes a debt to him today. I mean, that's a tribute to his abilities, but also his ethic of hard work. He would get in the lab at 4 a.m. He would beat me by half hour because I was getting in the lab at 4:30, and he would do that 7 days a week, which I would not do. And he just accomplished wonders. So we hit it off right away. There's Greg, there's the magnetic field, there's molecular expressions. One of the things of the many things he had done was he was also an entrepreneur. And so he put pictures of vitamins and cocktail mixes under a polarized light microscope, take pretty pictures, put them on ties, actually approached a guy who was selling ties and said, hey, you might want to do this. They did, and they made millions of dollars selling those ties. And that's what, among many other things, helped to fund Mike's lab. He didn't have to write grants. One of the other things is, again, he got bitten by the fluorescent protein bug, just as I did. And so he started pivoting to now doing a lot of fluorescence and a lot of live cell imaging work. And in order to be able to proceed with the different types of things he'd like to see, he started getting into being a molecular biologist himself and creating plasmids and for different fluorescent proteins. This was now the golden age of, there was a new fluorescent protein every month in some journal. And then you have to decide to tag that on every protein you're interested, check it out. So Mike had basically created an army of cloners who would do this work for him in his lab. And the people he picked were kids who had either flunked out of Florida State or who were in danger of flunking out of Florida State, just like he was in danger of flunking out before, and like he decided to leave Florida State from his PhD, and turn these kids around and show them that there's interesting things to do and prove to them the value of hard work. In the end, he mentored like 500 kids in the end. And so, but the one thing, the way Mike changed my life irrevocably was because when we went there and I was telling him about my optical lattice microscope, he was saying, well, you know, here's all these different fluorescent proteins. You might try to do this. Now there's this new type of fluorescent protein called an optical highlighter. So maybe you want to play with that. Now, optical highlighter initially does not glow unless you shine violet light on it, and then it will glow green. And so Harold and I started thinking about that. And then we were sitting, you know, we were thinking about, well, okay, maybe I can team up with Mike. Mike seemed reasonably interested in the idea of my building an optical lattice microscope and taking it to Florida State to work with him there. But then in the airport, we thought about this paper, which I published right after I quit Bell, which is there's a potential path to super resolution by the fact that all of your single molecules look like fuzzy blobs and they overlap at the diffraction limit. But if somehow they're different from one another, you can plot them in a three-dimensional space based on whatever that discriminating characteristic is there. It could be time, it could be color, it could be blinking rate, it could be anything. And so when we thought about this idea that you can turn on a molecule, well, I mean, we were just floored sitting there in the airport waiting for a plane. And we said, oh my God, if all we did was turn that violet light down low enough, we would only turn on a few molecules at a time. We could then find the centers of their emission and plot those, turn on another subset again and again and again, and get a super resolution image. So it was going to see Mike was really the key that went in the lock of this 10-year-old idea of how it would become practical in order to do this. And so we said, oh my God, this is so easy. Why hasn't anybody done it yet? And so we scoured the literature. Oh, there's nothing there. We got to do it. And it was easy enough to do. And so with that missing link, where did I go? I went backwards in time. Here we go. So now we got, so we did it ourselves in Harold's living. And so Harold had a lot of equipment from Bell Labs. He was able to take out of storage. We did put about $25,000 each for things like an EMCCD, which if you buy one yourself back in that era is a lot of money. And then we put it all together. And then we needed help in order to do it. Mike was willing to help, but what we really needed was that photoactivated fluorescent protein. So I had also contacted a buddy from Bell who was at NIH to see if I could do my optical lattice microscope there and give a talk there. So that talk was scheduled the week after we had visited Florida State. So it was all set up already for me to go there and say, hey, can you please ask Jennifer Lippincott-Schwartz and George Patterson to come to my talk? Because they're the ones who invented that switchable protein. And they did, and I took them to lunch and I said, my buddy and I have this crazy idea, can you help us out? And she said, sure. So, Mike was the perfect guy. I mean, I'm agnostic, okay? But if there's one thing to make me a deist, it's how in 1 month, all of these little dominoes fell over between seeing exactly the right person and the right time to have this all fall into place. Jennifer's whole career has been about being an early adopter of new technologies. And so many people would have blown us off as two unemployed weirdos, but she said, bring it by. And we did. And so April 2005 was Mike. Late April 2005 was Jennifer. By August, we had the microscope built in Harold's living room. September, we're doing this, 2005. So we're turning on the violet light so low with this protein Katie. And again, Mike was working to figure out, scouring the literature for any type of photoswitchable protein, and then getting his team to make plasmids for everything, for every different protein we would want to do with it. One of them was this one KD at the time, which worked pretty well. And so we turn on the laser light very low, we get the single molecules. If you sum up all those fuzzy blobs, you get a diffraction limit image. If you find the centers, you start to build the super resolution image. And after 20,000 frames, 50,000 molecules, you go from lysosomes that look like this to lysosomes that look like this, or better appreciate. So it was easy. It just fell right into our laps. And, at Bell Lab, at Janelia, the guy who founded Janelia talks about the deletion test. The only real metric that matters in science in the end is how would the scientific landscape change if X was removed from the equation. Well, I can say, at least in my own personal life, my own personal life would have been incredibly different, probably much less rich if Mike was not in my life. And I really owe him a big debt, and he was a great friend. So, but Mike's legacy goes way beyond just me. Molecular Expressions eventually became 3 different websites for the 3 major microscope companies. This is a resource that everybody I know still uses this day. I teach a microscopy class, and in that microscopy class, the number on the first day, the thing I say, you want to know anything about microscopes, you look at these sites, okay? And they do. He had a beautiful book on his imagery, and with Doug Murphy at Janelia, he put together all his website information in a nice book, which is also a required book for our course that we teach. And of course, his real legacy is those zillions of fluorescent protein fusions he developed, over 3,000 of them. We have a whole set at Janelia. There's sets at NIH, and of course, in Addgene, they have it as well for anybody who wants any of these sorts of things. And also, like I said, he's mentored 500 plus students over the years, made millions in royalties for FSU, and his plasmids have now been used in well over 600 papers. Pretty good for a guy who never got his PhD and was chroming parts not too long before, okay? So with Palm, we did many, many things, application examples. I said way too much about Mike, so I'm gonna skip over that. But this is the important message from my talk, which is, so I spent several years doing Palm, and I got very frustrated with it because I was looking at dead things, okay? It's very difficult to get the labeling density high enough. The gain versus pain threshold to me was not great of doing Palm. And so one experiment we did with Jennifer is, rather than trying to photoactivate every molecule in the sample to find out how it's decorating the specimen, let's just turn on in a live cell a fraction of them and watch their kinetics, watch how they move. So that became SPT Palm. And so this is an example where we're looking at proteins in the plasma membrane. When you have a protein that's known to cluster, you end up getting these little clusters in the diffusion map. When you have ones that are very mobile, you get these highly mobile tracks like that. in a nice experiment we then did with James Lewitt Genelia, we looked at the protein involved in Huntington's disease, where there's this glutamine repeat on top of it, which causes it to form these aggregates. These are the nuclei of, can we turn down the lights just a little? It's really hard to see that otherwise. When you have the wild type cell, they form tiny little aggregates, but they're very transient and go away. In the nucleus of a cell with the mutation, you end up getting these large aggregates. These have been known for a long time. Somebody's trying to find a happy medium, I think, somewhere. There we go. Okay. This one turned off for whatever reason, but basically the real surprise, though, was we did a 2-color experiment where we looked at different transcription factors. And what we found was that these aggregates are actually traps that suck in the transcription factors and keep them, therefore, from getting to the DNA. And so that potentially that could downregulate certain genes and lead to other phenotypes related to the disease. So it was a hypothesis generator in this case. So then we started working with Genelia, the Transcription Imaging Consortium, to look at these transcription factors in more detail. This is an animation that showed what was known at the time about how transcription works, where these transcription factors come to the start codon, and then eventually a polymerase is recruited and it scoots along the DNA and spits out RNA. So our collaborator, Bob Tejan, had, in 30 years of biochemistry, built up this idea about how these transcription factors come together at this initiation complex at the start codon to start the process. But then he wanted to look at this dynamically by SPT-POM, and what we found is none of these transcription factors bind to the start codon for more than a few seconds. And it was believed that this process would take somewhere on the order of an hour. So this is dogma. This is in molecular biology textbooks everywhere, but that's not the way it works. So what's going on? Instead, you have multiple copies of each transcription factor that kind of glom around the start codon like a bunch of bees around a hive. And what would cause them to stay there when they can move anywhere? Well, they have these intrinsically disordered regions on them that make them particularly sticky to themselves and partners. So it becomes sort of a stochastic yet sequential process of one transcription factor after another coming, recruiting the next until the polymerase is recruited. And so that completely overturned the idea of how transcription is done. And it led to the idea of Tej and Zavi, and Luke will be here later in the week, and myself starting a drug discovery company based on single molecule localization inside of live cells to see how the kinetics of the motion of these cells is changed under different pharmacological perturbations. And so we started this company in 2019, and it's been amazing how much they've learned about proteins and protein-protein interaction networks by doing this type of stuff. And they're really doing well. Obviously, it's going to be a while before there's a drug, but they have a number of candidates that have come out of this massive screen. They take about a petabyte a day and then reduce it in order to understand what's going on. So part of what I learned from that is that there's a lot to be learned by looking at live cells instead of dead ones. The other part that influenced me in this was we had recruited to Genelia Mats Gustafsson in 2008. And he was, of course, the world's expert on structured illumination microscopy, which by the fact that you use much lower intensities of light and requires only 9 images for 2D to reconstruct is much faster and less invasive than POM. And so he was doing beautiful work, but then he died of a glioblastoma in 2011. So I inherited Lin Shao from his group, and we wanted to continue working along those lines. So we did one project where we just went gonzo with an Olympus objective that has 1.7 NA for turf, and then did turf resolution at 80 nanometer resolution in the green channel. And you can do multicolors, you see there. We then also wanted to extend, he had done an early work on doing nonlinear SIM to go beyond that 2x limit SIM is normally bound to. but in that case, by doing some saturation. In our case, we decided to use photo switching again. So a guy in China, Pingyong Zhu, developed a photo switchable fluorescent protein, which looked promising. And so again, how do we get all the plasmids for all of this work I'm talking about? We go to Mike Davidson. He's our guy to do any custom work we want. Every paper we did for about a decade after Palm. Well, not a decade. No, he didn't live quite that long, but close. About 6 or 7 years after that. Mike was the guy we would go to get, Mike, we need this fused to this, fused to this. Couple weeks, boom, it's in the mail and back to us. So Mike was an author on all these papers. In this case, with the photo switching, we can get down to about 60 nanometer resolution. This is looking at early endosomes moving among the actin cytoskeleton. This is looking at how alpha-actinin bundles together actin filaments into larger stress fibers and so forth. We also did some work with just regular SIM, but now doing it. The trouble with turf, of course, is you're only seeing the bottom 100 nanometers at the basal membrane of the cell. So we did this sort of grazing incidence just outside of turf to look about 1 micron deep, and we could do very fast imaging up to a couple hundred frames a second to look at, for example, how the microtubules remodel the endoplasmic reticulum. These three mechanisms of sliding along the microtubules, attaching to the end of a growing end or attaching to a shrinking end were well known. We also found that the ER would hitch onto, for example, lysosomes, and lysosomes actively transported by the microtubules will move them along, or you would get de novo budding of different ER filaments. And we characterized their probabilities and how often and velocities. Surprisingly enough, we found on putting a whole Drosophila embryo, early embryo under it, that we could get close enough that 1 micron field would penetrate into these earlier amniocerosis cells on the embryo, and we could watch these pulsating contractions of actin. When it expands, you actually get little bubble-like things of the cells in the amniocerosa, and as the actin network contracts, it pulls it in, and so it kind of ratchets its way to pull those cells in, so that these other cells can come up and close the amniocerosis in time. No, let's see. Okay. So we did lots of sim applications. Again, I'm going way too slow, so I'll go over it. The one cute one here was with John Hammer's group at NIH. These actin monomers form these bipolar dimers, which together link together actin filaments to generate forces in cells. And we try to study this process of how they get together. What we found is that while you can get initial de novo formation of one of those dimer pairs from many, many copies of the monomers, the new ones form by actually splitting off. It's much easier for them to form by this sort of split, grow, split, grow. So you get this sort of cascade avalanche effect to then create all of the bipolar filaments inside of the cell. We also looked at the ER, and canonically, the ER at lower resolution consists of tubules and sheets. We showed that the sheets are nothing more than tubules that are moving too fast and are too tight, and there's separations for you to be able to resolve in either space or time normally, but with this high-speed grazing incidence sim we could. And then I was also motivated to move into live cell imaging because of the great work that Philip Keller and others were doing at Janelia with light sheet microscopy. And so the wrinkle we put on it, instead of using Gaussian beams, was to use a Bessel beam so we could decouple the spatial frequencies and the tightness of the central lobe in the light sheet versus its length, which is tied together with a Gaussian beam. And eventually that led to me repurposing my old optical lattice theory that we threw to the curb when we started doing Palm, and show optimal separations of these Bessel-like beams in a sheet to create what we call the lattice light sheet microscope. And then over the years, we've done a lot. In fact, I'd say since 2012, light sheet and lattice light sheet has been the dominant thing that we've done in our group, in part because it's popular. we're not a core ourselves, but for several years after developing this, we felt like a core because we brought in about 50 different groups into our lab to do this. Most of them go away with big smiles on their faces and 10 terabytes of data. And then they call up a month later and they're crying because they have no idea how to deal with 10 terabytes of data. And that's still a problem today, and where I'll end this talk is on that topic. So lots of applications over time. I won't go into this, but yeah, I have never had a microscope, including Palm, that has ever been as prolific as a means to see to reveal new biology as the lattice light sheet. So basically what I found after first doing years of work with near field microscopy on dead stuff or things that were never living to begin with, like circuits, what have we learned? With live cell single molecule tracking, with SIM, and with lattice light sheet, all biological structure and physiology arise from dynamic processes occurring at spatial scales, emergence from all levels, from single molecules, basically up to the entire biosphere, okay? Second thing is that the one thing there's lots of competition when it comes to structural imaging, but the one thing optical microscopy uniquely has is the ability to observe these dynamics in living systems. And so still to this day, despite all of the work we do in imaging, most biologists, speaking as a professor in the Department of Molecular Cell Biology and looking at what my colleagues talk about and do, most of them still view biology by these incredibly successful paradigms of the 20th century: biochemistry, molecular biology, and structural biology. The problem is that they're all reductionist approaches that involve taking the cell apart and trying to look at its pieces. Well, if you're going to look at a dynamic system like an internal combustion engine, it's pretty damn hard to reverse engineer from a random pile of engine parts, which is basically what 20th century biology is about, okay? Is to try to understand, but you don't have the right stoichiometry, so you don't know how many, if you have the right number of gears, you don't even know if you have all of the components, and you don't know how they interact. And so it's, I think, a fool's errand to try to get to a complete understanding of any dynamic system. And you're talking now about a dynamic. One cell has several billion molecules, 20,000 different proteins. It's the most complicated piece of matter we know in the universe. It's really daunting. And so animation and natural variability are the fundamental hallmarks of life. It's the thing that defines life, is animation. And so all these biologists working on those other approaches are ignoring these elephants in the room. And so how can you understand life if you don't look at it live? Okay. So this has been my mantra since about 2012. The problem is I've learned enough biology that cells didn't evolve on cover slips. And we know that the phenotypes that we observe are the result of gene expression. And we know that gene expression is controlled by the local environment. So if you don't have the proper local environment, how can you trust the phenotypes you're seeing in the microscope, no matter how good your microscope is? And so we need to take cell biology away from the cover slip. So yeah, this is starting to get a little iffy. I may have to reboot in a little bit, but let's just look at this little guy. So people have been looking at subcellular dynamics with 2-photon and other techniques. This is, for example, looking at a neutrophil moving inside a mouse lung in this case. But I think you would agree that the resolution here isn't quite what I was showing in those lattice light sheet images. So what we need to do is we need to somehow deal with the problems of aberrations from heterogeneous tissue when we try to apply lattice light sheet inside a whole organism. So as usual, I give a talk in which I say we biologists are the retarded stepchildren of astronomers because we always steal everything from them 50 years after they do it first. And so another case is adaptive optics, where they shoot a laser into the stratosphere to create an artificial guide star, which is then used brought back into the microscope, into the telescope to find out how the atmosphere is aberrating the light. And then they put it off a deformable mirror to change the shape and then get boing back to this or boing back to this. So this one was actually related to the Nobel Prize a few years ago, looking at the supermassive black hole of stars orbiting at the center of our galaxy. So as you can see from this video, the potential gains or I won't say gains, recovery of resolution is pretty dramatic. Yes, so, okay, now we're really, okay, let's do a reboot here. Silly. Vanessa, can you give me a hand to just get out of the program first? Yeah. There, perfect. Now just go up to, go here. One more up. There we go, perfect. Appreciate it.
Of course.
Good, okay, well, wasn't so bad. All right, I'll try to go quickly here. So now this is combining the adaptive optics with the lattice light sheet. Oh, yeah. Good. All right. So now we can combine those things together. So this is an example of looking at the development of the spinal cord inside a developing zebrafish with sort of a brainbow type technique called autobow, so that each neuron is colored in a different color. This is now, you saw that earlier picture of a neutrophil. This is now a neutrophil in the ear of a developing zebrafish. It just scooted there. You're seeing a fibroblast cell that just went through mitosis. These are the brain cells underneath here. These are the skin cells at the top. This is looking at 3 stages of the extravasation, towards extravasation of a human breast cancer cell injected in a zebrafish as it goes through the capillary and starts to come out. This is showing early development near the eye of the fish. This is still probably my favorite slide ever, because it shows how beautiful life is and how crazy, dauntingly complex life is, and how much work we still have ahead of us to try to understand this. So the big problem, though, is now you've got high resolution, several hundred nanometers in all directions. Now you're looking at whole organisms, so you're covering hundreds and hundreds of microns in each direction, sometimes millimeters. You can guess that's a lot of voxels, right? A lot of voxels. And so this is the problem. But what can we observe? Again, like I say, anytime we're drawing conclusions from cells on cover slips, there's an asterisk behind them. And I've seen over and over again in tissue how ER looks different, how mitos look different, how the cytoskeleton looks different, in different tissues in different cell types across the organism, different from a cover slip. So to what extent are the morphologies and process observed in culture cells recapitulated in multicellular organisms? How do they differ? I know they differ. We want to characterize it. What's the distribution of variability? Variability is a hallmark of life. It's the fact that we have this variability that makes life tick. What are the native distributions? How are they affected by pharmacological or genetic perturbations? How do they manifest them? How do subcellular events manifest themselves in collective behaviors of cells and tissue? All of these questions are potentially addressable with our lattice light sheet with adaptive optics, but we have no idea how to analyze that data. So after our initial train wreck lattice AO, we decided to make a 2nd generation instrument that you see here. And because it uses 2-photon laser, a bunch of linear lasers, deformable mirrors, etc., we made it into a Swiss Army knife that can run in all these modes. But I won't go into that in this talk. This is what we call mosaic. Here's a couple examples. This is looking, guess where these cells came from? We did this imaging a year and a half ago. Those are Mike's cells. Mike made some stable cells, in this case with ER and histones. We use those as test cells all the time. So this is practically a millimeter square that we're looking at every 90 seconds, because this is a super performative microscope. It takes 4 terabytes of data an hour. This is running it flat out for 10 days at looking by expansion microscopy at the mouse olfactory bulb, and it's 500 terabytes of data in 10 days. Well, we had the first step in order to be able to analyze data at this scale and at this speed is that you have to be able to do the pre-processing in real time, because nobody's going to have enough hard drive space to do this ex post facto. So we developed a pipeline with our own cluster to be able to do all of the deconvolution, all of the rotations, all of the stitching of multiple tiles, all of the compression to put it on a hard drive, all in real time. And that's what made these things possible. This is just got accepted in Nature Methods. We've also worked with NVIDIA, who has this multi-GPU solution to be able to do real-time imaging so that you're not flying blind when you're taking 500 terabytes of data, but can know what's going on, to be able to look at the data as it comes out of our real-time processing pipeline to do sort of real-time visualization. But the big problem still is then trying to interpret all of that data once it's been stitched and put together. In the past, we've done this by just essentially custom coding solutions to look at different biological events, such as how nuclei are different in different types of cells and development and so forth, or how the myelination in this expansion microscopy example changes between different pyramidal neurons as you go to the white matter. So both of these papers were largely done by my friend and really at this point my twin, Gokula Padahayalua, who's running our Advanced Bioimaging Center at Berkeley, but is also deeply involved in everything that I do. So when he was working in Tommy Kierkhausen's group, this paper, to get all the insights we had, 3 million CPU hours in 1 year of his time. For the expansion paper, another 3 million CPU hours and a year of his time. That's not a scalable solution because I don't know how to clone them. So we need a better answer. And the problem is that even in a very tiny piece of tissue, the number of questions you can ask or the number of things you can see are enormous. This is just one small data set where we cut the tail of a developing zebrafish and then started imaging histones and cell membranes. And you can look at mesenchymal cell dynamics, you can look at how immune cells move, you can look at how the blood vessel, the capillaries actually remodel over time in response to the cut. You can look at how they anchor, the epithelial cells anchor to the low. There's a million questions you can ask, and each question is going to take one human some number of months in order to quantify. So how do we get around that? Well, you know, I've watched the development of AI over time and have never been terribly impressed, and generally unimpressed by its application in microscopy. So you're talking to a guy who's been a skeptic about it, but who's come kicking and screaming to believe that AI may be the only way to be able to have scalable image analysis of what we want to do. So you start with a beautiful piece of imaging where you can see several different organelles inside a hunk of tissue, but how do you interpret that? It's just too damn complicated. Well, one of the big breakthroughs, I think, was with the large language molecules like ChatGPT. And the big advance there was the self-attention, basically developing an AI that looks at the sequence of words that then allows it to predict words in a sentence. Well, basically, you're looking at spatio-temporal priors in order to do that. Well, spatial-temporal priors and understanding spatial-temporal relationships is what imaging is all about. And so, in fact, you know, Google was able to take these transformer models and use it to then be able to classify different images automatically inside of 2D images, different subfeatures, by pre-training it on 300 million images scraped off the web, 45 terabytes total. They thought that was big. We laugh at 45 terabytes. To us, that's an afternoon, okay? So one of the things about these models, they require vast amounts of data for pre-training because there's no scalable way to do annotations on all this stuff to train the model. So it has to use pre-trained data, but it requires vast amounts. Well, we're really good at taking data, so that's not a limitation for us. So Meta used this vision transformer that Google developed, and then fine-tuned it, so to speak, by using annotated data to do fine-tuning for segmentation. So when I looked at this, I said, well, the first step of taking that block of tissue is to be able to segment those cells in 4D, okay? And so, so this is a potential avenue. So you want to be able to do this, okay? GoCool did this by a lot of manual work and a lot of custom code, but ultimately you want this to happen seamlessly and quickly with an AI to be able to separate the cells and look at them individually. In our case, what our goal is take a lot of pre-trained data on a whole bunch of zebrafish looking at membrane, and then after that do fine-tuning with the annotation for the fine-tuning for segmentation done by the fact that biology will already do that for you with something like Brainbow, stochastic multicolor labeling, and use that as the means to get the annotations to do that. And then once we have it all, these cells separated, now you've taken cell biology away from the cover slip. At this point, you can do what you do today. You can pick out an individual cell, do whatever type of analysis, look at it, understand it, because you can pull them apart. But we want to go much deeper than that. We want to be able to automate this process of finding the spatial temporal changes inside of cells over time and how they relate to other cells and so forth. So you could figure out cell types, cell states, classify the organelles, look at the interactions, look at how things remodel, correlate them to other orthogonal data in what they call, basically you develop a foundation model that tries to pull together different types of information inside of a transformer to look at the relationships. And ultimately, because, I've worked as hard as any of you guys to try to get my tools in the field and get my tools used, and it's always a struggle. It's always a struggle to get people who can use the tool, continue to use the tool. If any of you heard, I gave a rant in February that's on YouTube about this whole thing. And what I think is critical for this thing is to be able to then put a large language model around it. So it would be just like, just ask the model, how does the nucleus of a cell have to change when it ends up trying to extravasate through a blood vessel? It would pop up 12 movies from the 25 petabytes of data it has, show you those exact events, giving you some curves about exactly what happens to, the morphology of the nuclei, so forth and so on. And you could ask literally billions of similar questions. It would be just like what happens when cell biologists are together and just, hey, what if, what happens when, and you could just query the model and get that directly. So I know that sounds like science fiction, but that's the direction which we want to head. And it's going to require a huge number of reagents and lots of transgenic fish. If only Mike were alive today, then he could help us out with this, but we're trying to do the best we can on ourselves. It also will require vast computing resources, probably bigger than anything that's been tackled in AI so far. A lot of my time now is actually going on the road and talking to AI people and trying to get them excited about this vision. Ultimately, we'd like to create something like what we call a cell observatory, where people could even put in proposals to basically ask their question. We would take the data for them because the microscopes are complicated, the data analysis is even more complicated. Basically, they would, just like an observatory now, no astronomer goes out with his own telescope in the backyard and takes data. He goes and he puts in a proposal at an observatory and gets it. It would be that same kind of model. And so finally, to close, this is probably one of my favorite pictures of Mike. It's kind of bittersweet because, when I met Mike in 2004, he's a guy who burned the candle at both ends, as you can imagine how hard he worked.
And as a result, like at times I would burn the candle at both ends. And what happens with me is I put on weight, okay, and I get, just all amped up. And Mike, he smoked for many years. He worked like hell. And a few years, I think, before his cancer diagnosis, he said he realized something had to change. And Mike being Mike, when he decides to do something, he does it. And so this is the lean and mean Mike that you're seeing here, before his cancer diagnosis. And so he was a guy, he really believed that anything he set his mind to do, he could do. And I've really looked up to him my entire life. I still believe in the value of hard work. I think hard work trumps everything. Everything we do is in science is first about fumbling around. We don't know what we're doing when we're doing something new until we do it. And the only way to work your way through the weeds and finally start to feel a direction is by putting in the hours in order to get there. And I'm going in now in yet another new direction with this AI, and it's going to be a big, hard effort. One of the things we need is we need more help. So if you guys know anybody, if you're interested yourself or you know anybody else who would like to come work with us on this project to try to do this AI-based AI-based discovery, and work on a mosaic and help us take the 25 petabytes of data we're going to need, I would be happy to talk to you. And again, I just want to say in conclusion, Mike, wherever you are, thank you for everything. You changed my life, and I still look up to you as my mentor. Thank you very much. Yeah, sure.
Well, thank you very much, Eric. And I'm really glad that we're in a movie theater for this because I feel like it's a completely appropriate venue. All we needed was a little popcorn.
There you go.
So I'm Liz Haynes. I'm an incoming assistant professor in the Department of Cell and Regenerative Biology here at UW-Madison. I'll be handling questions for this session. If you have a question, just raise your hand. I'll bring you the mic.
All of these are difficult questions as you get up in scale. When the scale is small enough, what we do right now, for example, as part of collecting our data for this sort of AI mission, all of the data goes into a Google bucket as soon as it's been pre-processed, and it has metadata along with it. But again, even trying to do that, navigating that for most people is just going to be next to impossible. I don't have a good answer for that. For us in our model, we know what to do with it. In terms of the, keep it or throw it away thing, I think there's a level, that's always a really tough decision because we take so much data. It's always very painful to say, when do we triage something, it would be nice to keep it all. I would disagree with you about astronomers. Some of the data they take is so voluminous, they don't save it after a while. They have to triage it. And then again, you just retake it. That's in much of the data sets, if they're large, and once we've gotten the value out of reducing what we think is there, we'll keep representative samples of the data, but we do have to triage the vast majority of it because it's just too much. And it's, taking the data for now at this point is the least challenging part, or the one. Not to say that the guys who run the microscopes don't have to work very hard to get quality data, but it's something we know how to do well. Okay. Yeah, but I understand from most people don't have the resources we have. Most people don't have the cluster we have to be able to do the kind of, doing this sort of real-time ability at 4 terabytes per hour to analyze. So yeah, you have to make just rational decisions based on the resources you have. The other problem that bothers me a little bit, Janelia has also been very much about collecting massive data sets like Flylight, the Fly-EM Connectome, and other things. And what the metrics people normally use is how many hits their portals get for this stuff. But there's a huge difference between hits and use. And there hasn't been a lot of use considering the amount of effort and money and years that have gone into it. So this is another big disconnect that I don't think has really been properly appreciated yet. Yeah.
Please introduce yourself before you ask your question as well.
I'm Megan Driscoll. I'm an assistant professor at the University of Minnesota. And I love that a large language model would make the data more accessible. I mean, but we also already have a language for dynamics. We have a mathematical language for dynamics that's been
Yeah.
developed over centuries. So how do we integrate that language, right? The language of geometry and physics into our analysis without making the analysis too hard to accomplish?
Yeah, I don't know. Ultimately, I agree that there are representations of the data, particularly dynamic data, that it can be useful, but they will be useful for people trained to understand those representations like yourself. In the end, again, I'm really, really concerned about building it and them not coming, because I've done that many, many times in my life. My feeling based on my experience with collaborators has been the best way for them to be able to interface with the data is first through a verbal discussion, followed by a visual representation of the things that they're asking about, followed then by, well, let's look at this one closer, that kind of thing, right? So I'm just trying to think about the human factors involved, right? And so that's what I'm trying to get at with the LLM in the end, is certainly, obviously, this has been a big hit with the general public in terms of getting them to be able to access information, both with, LLMs that they can query directly or use themselves. There's obviously huge dangers in it too. But I think this is at least a possible path forward. Either that or I should go with sailing or something instead of continuing on, but yes.
My name is Zhao Jie Zhang from University of Wyoming, different UW.
Yeah.
It seems to I do a lot of electron microscopy work, but it seems to me that your work is giving a death sentence to electron microscopy.
Absolutely not.
So my question is that, in your view, what is the future for electron microscopy, if there is one?
I mean, you know what my best friend Harold does these days? He does volume electric microscopy with FibSim, okay? He's a god in that building because of it. It was because of his microscopes that they were able to get the entire fly connectome, which has been a dream of neuroscientists for 50 years. They have this cell map project where basically they do AI to segment out stuff in different tissues and so forth. There's I got into microscopy because I believed resolution was the metric that mattered. It is a metric that matters. It still matters. And no matter what we do with Palm and so forth, there are things that you can see Harold and I spent 2 years doing correlative EM Palm because of the understanding that neither gives a full picture and that they're very complementary. Crazy difficult project, by the way, but again, yes, everything is complementary to everything else. EM has resolution, of course, AlphaFold, with cryo-EM and then crystallography being brought. That's the big biological hit of AI so far, right? The only biological hit, in my opinion, of AI so far, AlphaFold. It's a big deal, okay? And it wouldn't happen without cryo-EM. So there's a lot to do with EM, but I'm firmly convinced that the reason I'm so strident about saying about live imaging and pissing all over everything else in biochem is because people don't understand the value of live imaging. And it seems so obvious to me in time by looking at enough live things that it's going to be that the real frontier, the area where I think the big gains will be made is going to be in live imaging. And I think we're poised to do that, but a lot of people still have these blinders on about structural biology, which has value. But one of the other things about my career is I always try to find a place, let everybody else march in this direction. I'm going to march in this direction. Okay. And I think, that's kind of where live cell is today.
Hi, my name's Kristen. I'm at the Van Andel Research Institute out in Grand Rapids,
Oh,
Michigan.
cool.
I'm just wondering I do image analysis
Yeah.
as part of my career, and thinking about like the pre-processing that you're discussing, like that
Yeah.
obviously requires a lot of resources,
Right.
and trying to make that more accessible to places that might not have those
Right.
kinds of resources at hand.
Yeah.
I'm just kind of wondering, like, what would you imagine would be like an ideal setup to make that kind of stuff more accessible for
Yeah.
smaller institutes that don't have those resources? And then a secondary question to integrating the language, the LLMs
Yes.
into analysis. I know as far as like using things like that for my career,
Uh-huh.
it can very strongly hallucinate things
Sure.
very confidently.
Yes.
And wondering how we should think about training that kind of model to actually make sure that we are getting accurate data if we're going to use something like that to query these really large data
Yes.
sets.
Good questions. Okay. So the first one about the processing. So that pipeline that I said that is going to be published, it actually seamlessly adapts to however much resources you have. So you can use the same pipeline if you have one GPU and one workstation. Obviously, it's going to take a lot longer, but then maybe you don't take data at 4 terabytes per hour either. So we've been worked very hard. In fact, the Nature Methods editors kept our feet to the fire, as did the reviewers, to be able to show that on very limited hardware that it's still performative. So once, with that stuff, if you go to the ABC website, you'll see it, but it'll also be available. It's also bioRxiv. There's a GitHub repository, et cetera, already. So, you do what you can with the hardware you got, and we tried to make a solution that would allow people to do with what they want. Hallucinations and LLMs. So yeah, that was my big beef when I see a lot of super res AI where they're imputing super resolution from whatever. Whenever I saw it, I would say either, well, A, that's either obvious, or else B, that's clearly erroneous, because people would take diffraction limited images of ER or microtubules or something and show me the super res thing. And I've looked at plenty of microtubules in ER by super res, and I know when I'm seeing shit, and a lot of times I'd see shit. Okay. And so I'm totally in the hallucination camp. I think a big part of trying to rely on AI for what comes out is, again, through the LLM, to be able to see very easily by your question, to see the raw data, to see the pre-processed data, and to see the AI mucked around with data all at the same time. Okay. So I think that's how you'd have to go. But yeah, it's a Pandora's box, and there will be lots of people who will not be paying attention to what demons are flying out of that box, but I don't know what to say. Yeah.
Real quickly, there's, really great solutions out there for dealing with vector decks with adaptive optics, and of course, clearing from fixed tissues.
Yeah.
But scatter, as you know, remains a massive
Sure,
challenge in life.
absolutely.
So what is your vision there? Where do you
I
see
don't
that
have
going?
I wish I had a good answer on scatter, but I don't. So, obviously you saw that recent paper that just came out with the Doritos dye. There's lots of caveats with that one too. But yeah, basically our answer is to go as deep as we can with the scatter. We can do some 2-photon light sheet with some filtering to get the ballistic part, not the scattered part. But yeah, there's a lot of glory to the guy who can figure out the scattering problem.
All right, this is Vanessa again. We have a question from the audience from Jose Moreno, who's a postdoc at Albert Einstein College of Medicine. Said we're developing a hyperspectral microscopy and for transcriptomic solution. My question is, when bringing new microscopy technologies into the field, what hurdles you face to get them adopted into the mainstream?
Yeah, it depends on how you want to do that. If you bring people to your own lab, there's the resources that you have to bring to bear. For us at Janelia, the development of the Advanced Imaging Center there was a huge benefit. That's what got us away from, I told you we were an imaging core and work with 100 different groups on Lattice Light Sheet. Well, the thing that finally got us away from that was hiring Leong and getting that imaging core. Yeah, it's I don't have really good answers is all I can say. It's really a case by case basis as to how you do this and find out what works. Yeah.